Now, looking at neurogenesis and gliogenesis. After patterning, the neural tube's which is lined by neuroepithelial cells, undergoes rapid proliferation. These cells are multipotent progenitors that give rise to neurons and glial cells. Neurogenesis involves neuroepithelial cells first differentiating into radial glial cells, which serve as the progenitors for neurons. Neurons are generated in a highly regulated sequence, with different classes of neurons being produced at specific times. Newly formed neurons migrate away from the ventricular zone along the processes of radial glial cells to reach their final destination in the brain and spinal cord. Following neurogenesis, radial glial cells begin to produce glial cells, including astrocytes and oligodendrocytes in the brain, and maintain the blood-brain barrier, while oligodendrocytes myelinate axons in the central nervous system, which facilitates rapid signal transmission. Here, conditions associated with neurogenesis are micro and megalencephaly. Microcephaly is when there's a reduced brain size due to impaired neurogenesis, often from genetic causes, which affects neural progenitor cells. Microcephaly is associated with intellectual disability and can increase the risk of epilepsy. Megalencephaly is the opposite of microcephaly, where there is an overproduction of neurons, leading to abnormally large brain. This condition is linked to epilepsy and other neurodevelopmental disorders. Neurogenesis is also heavily involved in temporal lobe epilepsy, as the hippocampus is dependent upon neurogenesis for the formation of new memories. However, abnormal neurogenesis can really disrupt neuronal networks within the hippocampus and other temporal lobe regions, and is often associated with temporal lobe epilepsy. Tuberous sclerosis complex is another genetic disorder that leads to the growth of benign tumors in multiple organs, including the brain, again due to mutations affecting cellular differentiation. TSC is often associated with epilepsy and other developmental delay and autism, as it's involved in the mTOR pathway signaling. Migration and the layer formation. Neuronal migration is a critical phase in the CNS development, determining the architecture of the brain and spinal cord. Migrating neurons use radial cells as scaffolds to reach their destination. The cerebral cortex is formed from the migration of neurons in an inside-out pattern, where early-born neurons settle into the deeper layers and later-born neurons migrate past them to form the superficial layers. This process is essential for establishing the layered structure of the cortex, which is crucial for its function. Cerebellar development includes granule neurons migrating from the external germinal layer to the internal granule layer, while Purkinje cells settle in the single layer, forming the characteristic cerebellar circuitry. Here, lysencephaly is a severe neuronal migration by a smooth brain, which is lacking the normal folds and grooves known as gyri and sulci. It results in severe developmental delays, muscle spasms, and epilepsy. Lysencephaly is often caused by mutations in specific genes known as LIS1 and DCX. Polymicrogyria is another condition where the brain develops too many small gyri, leading to abnormal cortical layering. Polymicrogyria is associated with epilepsy, cognitive impairment, and speech and motor deficit. This results from the gyri, which are the infoldings of the brain, not being properly formed, and instead of having big, deeper grooves, they have many small, shallow grooves. These gyri are crucial for setting up the highways of information and arranging the cortex and its architecture in space for its functioning, and also providing the complexity of neuronal networks, and these infoldings really give the brain its complex structure for its function. Axon guidance and synaptogenesis is the next thing we'll discuss. As once neurons reach their final locations, they must extend their axons to connect with target cells to form functional networks. Axons are guided by a combination of attractive and repulsive cues in the environment, mediated by molecules such as netrins, semorphins, ephrins, and slits. Growth cones at the tips of axons respond to these cues, steering the axon towards its target. Once axons reach their target, they form synapses, establishing the connections necessary for neural circuits. Synaptic specificity is achieved through the interaction of pre- and postsynaptic adhesion molecules such as neuroligands and neurexins. Here, the formation of synaptogenesis is crucial for the communication between neurons and setting up functional networks. However, malformations in these can lead to a range of disorders and are especially linked with epilepsy. Kalman syndrome, a condition caused by defects in axon guidance, particularly affecting the olfactory and gonadotrophin releasing hormone neurons, leads to anosmia, loss of smell, hypogonadotropic hypogonadism. This is due to the failure of these neurons to reach their correct destinations. Autism spectrum disorder is increasingly understood to involve abnormalities in synaptogenesis and connectivity. Dysregulated synapse formation or pruning can lead to characteristic social and communication deficits, as well as the repetitive behavior seen in ASD. Synaptic dysfunction is a key feature in many forms of epilepsy as well, for instance in mutations in the synaptic proteins such as neuroligan 3 and neurexin 1, which are linked heavily to epilepsy and autism spectrum disorders, which suggest shared pathophysiology related to synapse formation and maintenance, which is often why people with autism have an increased risk of developing epilepsy later in life.